Hi there. Welcome to AV Biofun with Dr. D. This is part 2 of your unit 3.1 to 3.4. In the first part, we talked about free energy and ATP, the ATP molecule, the free energy carrier. In this part, we're going to talk about metabolism and enzymes. Now, I know you have a pretty good background in enzymes, but here we're going to focus on some key concepts. Enzymes are proteins. That is, the majority of enzymes are proteins. Some, rather few of them, are actually not proteins. They're RNA molecules and they're called ribozymes. But we're going to be focusing mostly now on proteins. Enzymes are catalysts. That means they're substances that speed up reactions without being changed, without being altered, without being consumed in the process. Enzyme names usually ends in ASE, and usually enzyme names contain the name of the substrate they use, but not always. Now, it is important to understand, to remember this key concepts. Enzymes do not alter the free energy of a reaction. They do not affect delta G. Enzymes cannot determine whether a reaction is spontaneous or not. All they can do is change the rate at which the reaction occurs. And let's introduce another concept with which you should be familiar with: activation energy. That is the energy to get something going, to get a chemical reaction going. A good analogy would be that initial energy that you need in the morning to get up at 6 o'clock after you've gone to bed at 3 a.m. Once you get up, once you have overcome this energy barrier, you'll be ready to go. You'll have breakfast, you'll get dressed, you go to school. But that initial burst of energy you need to get out of bed when you haven't gotten enough sleep, that would be a good analogy for the activation energy. Activation energy is an energy barrier that must be overcome by molecules in order for them to participate in a chemical reaction. And that activation barrier is present whether the reaction is spontaneous or not. If the energy releasing reaction, in an energy releasing reaction on the right, the reactants have more free energy than the products, the reaction is spontaneous. Still, there's an energy barrier here at the top of this graph. And the difference between the top of the graph, that energy level, and the free energy of the reactants is called an activation energy. In an energy absorbing reaction to the left, the reactants have less energy than the products. Still, that top of the graph, the free energy of the top of the graph is greater than that of the products. And the difference between the free energy here, the top of the graph, and the products is the activation energy. And we're going to explain in a minute what that top of the graph represents. So enzymes, what they do is they lower the activation energy without changing delta G. Again, the difference in free energy between reactant and products is not going to be altered by the enzyme. What is altered is the activation energy. So here's an analogy. Here's the energy level of lactose. Here's the energy levels of glucose and galactose. So this is an energy releasing reaction, a spontaneous breakdown of lactose into glucose and galactose. Lactose, the sugar in milk. However, before lactose can be broken down into glucose and galactose, that man here needs to push the ball, lactose, to the top of the hill before the ball can now roll down the hill spontaneously. And the difference between where he starts and the top of the hill is really the activation energy. What an enzyme will do, they would flatten the up curve, the uphill. So the person will need to climb less of a distance to go to the top of the hill, but the distance between the top of the hill and the bottom of the hill will be the same. So the reaction will proceed with net release of energy. So a fit, this is another thing that is really a key concept that you need to understand and remember, that the fit between an enzyme and its substrate is precise. The substrate will bind to a specific region of the protein of the enzyme called the activation site. And here I have provided you with a link to a flash-based, unfortunately it's flash-based animation that explains how enzymes work. I'm not going to play this, but for a few more weeks or maybe months, hopefully, you will be able to work with this animation. This animation is still working, even though it's flash-based. So what is activation energy? By definition, the activation energy is the free energy of the transition state. The minimum amount of energy that reactant molecules must possess to reach a transition state. So what is a transition state? Reactants need to reach an intermediate chemical state called the transition state. At the transition state, an unstable intermediate is formed. The transition state always has higher free energy than that of the initial reactants, and also higher energy than the products. Here are the reactants A, B, and C, D. In the transition state, they have formed unstable compounds, unstable intermediates. And these intermediates are so unstable that they'll proceed spontaneously towards forming products. The rate of a reaction is always dependent on, it's always proportional to the fraction, the proportion of molecules with energy equal to or greater than the activation energy. So the molecules will never be able to react unless they have energy equal to or greater than the activation energy. The more molecules we have that have that energy, the faster the reaction will proceed. The fewer molecules we have, the slower the reaction will proceed. Only molecules that are able to react at a given time are those with enough energy to exceed the activation energy barrier. So how can we change that? Let's look at this graph. This is a histogram. On the y-axis, we have number of molecules. On the x-axis is the kinetic energy of those molecules. So at temperature 1, let's say temperature 1, T1, is the body temperature of a human, 37 degrees Celsius. And here is the activation energy needed to get a reaction going. So look at the black graph. At this temperature, only a tiny little portion of molecules have energy equal to or greater than the activation energy. So the reaction will proceed very, very slowly. What if we were to raise the temperature? Now, we can't do that without killing humans, but theoretically, if we were to raise the temperature of the system, T2, now molecules will have greater kinetic energy, and there's going to be a greater number of molecules possessing energy equal to or greater than the activation energy. So the number of molecules at T1 at normal temperature that have surpassed the energy barrier is tiny, N1. But N2, the number of molecules that have surpassed that energy barrier is much, much greater. So the reaction will proceed much faster. Now, unfortunately, we cannot raise our temperatures. It will. And we cannot raise our temperatures to make reactions happen. So what we do, living organisms, is instead of raising the temperature, we're simply going to be moving that barrier. So here's the initial barrier, the energy barrier. We're just going to move it to the left. So when an enzyme is involved, an enzyme will lower that activation energy. It will lower that barrier. So now we have a greater amount of molecules that have energy that is equal to or greater than the activation energy. And it's represented here again. Y-axis is free energy, x-axis is progress of the reaction with time. So without the enzyme, this is how much activation energy you need. With the enzyme, you need a lot less compared to black arrow to the red arrow. So the enzyme will lower that activation energy, which means the reaction will proceed a lot faster. Now, most chemical reactions in living organisms have pretty high activation energy. And very few molecules can exceed that energy barrier. Life is really dependent on high activation energy. And we discussed this in class, why that is. if the activation barrier wasn't so high, chemical reactions will be happening all the time at high speed. There is no possibility to control chemical reactions. Resources will be wasted, energy will be wasted. Maybe products that are toxic would accumulate. Maybe there will be not enough reactants. There's many different consequences to chemical reactions gone haywire. So if most chemical reactions have a high energy barrier, that means they will not happen unless we add an enzyme, and enzymes can be controlled, they can be regulated. This gives the ability of living organisms to control, to regulate, to fine-tune chemical reactions. So enzymes are biological catalysts. So all catalysts really share three basic properties. They increase reaction rates by lowering the activation energy required. They form transient means temporary, reversible complexes with substrate molecules. And they will change the rate at which equilibrium is achieved, not the position. That means they do not change delta G. Remember, enzymes do not change delta G. Enzymes will not change whether a reaction is spontaneous or not. Enzymes cannot tell you whether the reaction will happen or not, with or without energy. Enzymes only speed the rate at which the reaction proceeds. Organic catalysts are enzymes. Active site is where the substrate binds. Now, in biology, you most likely learned that the substrate and the enzyme fit like a key and a lock. So there's a perfect fit between the substrate and the active site. In reality, it's not really a key, more of a key lock. It's an induced fit model. That means once the substrate binds to the active site slightly changes the shape so that it'll perfectly encompass the substrate. The enzyme's active site is still highly specific for the substrates, but the binding of the substrate to the active site, it's almost like Play-Doh. The enzyme active site molds itself around the substrate. This is the so-called induced fit model. So because of this precise chemical fit, the active site of the enzyme and its substrate, enzymes are highly specific. You have an enzyme regulating the rate of a chemical reaction, one chemical reaction, not two, not three, not four, not five, one. So each step in a metabolic pathway is regulated by a specific enzyme. Now, which amino acids do you think would be involved in being part of the active site and binding to the substrate? And I'm going to remind you that what I just said, the substrate binds temporarily to the enzyme. If it's temporary, if it's transient, these links are not covalent, they're not permanent. They're usually weak interactions. So of the 20 different amino acids, only few are actually involved in the active sites. Few meaning 6. These are the 6 amino acids that can participate in substrate binding. They're part of the active site. Why these? Can you guess? And I already told you, the amino acids of the protein of the enzyme, the side chains, bind to the substrate using weak bonds temporarily, not permanently. So once at the active site, the substrate molecules are bound to the enzyme surface in the right orientation to facilitate reaction. That binding usually involves hydrogen bonds, remember those are weak, ionic bonds, or both. So these amino acids are either polar, so they can form hydrogen bonds, or charged negatively and positively, so they can form ionic bonds with the substrate. The binding of the substrate to the enzyme is readily reversible. So the substrate really needs to be activated. Once it binds to the enzyme, it's activated. The role of the active site really is to recognize the substrate, to bind it, and also to activate it. Well, how does it activate it? It provides the right environment for catalysis. So instead of two molecules, let's say the reactants are two molecules, they need to meet and bump into each other to react, to form a chemical reaction, the substrate binds them together. It brings them together. So it brings two substrates together. If the reactants is a single substrate, then the enzyme binding to that single substrate can change its shape in such a way that makes it unstable and it makes it easier to break its bonds. And this is called substrate activation. This is how enzymes function as catalysts. This is how they lower the activation energy. This is how they speed up the chemical reaction. The sequence of events illustrated in this figure is with the enzyme sucrase. Sucrase comes from sucrose and ase, that means breaking down of sucrose. Sucrose is table sugar. Sucrose, which is the substrate, will bind to the active site of the enzyme. The active site of the enzyme is going to slightly change shape. The R groups of the specific amino acids at the active site will form weak bonds with sucrase. The second thing that will happen is that water, which is the second substrate, will bind to the active site. Then the substrates are converted to products, right? Hydrolysis reaction, glucose and fructose are produced from the breakdown of sucrose. The products will leave the enzyme, the active site, and the active site will be restored to its original shape. And this is a continuous cycle, and this illustrates the most important point of all. Enzymes are reusable. An enzyme molecule will bind a substrate, convert it to a product. Once the product leaves the enzyme can bind another substrate molecule. Once that substrate molecule is converted to product, it'll bind another molecule and another. Why? Because enzymes are not altered, they're not changed, they're not consumed in the reaction, so they can be reused multiple times. And this is the end of our first tutorial on enzymes. And next class, we're going to be talking about how the environment affects reaction rates in enzymes. Until next time.